Before we examine the Singer-Nicholson model or the fluid mosaic model, it is useful to go back and look at where did this model of the cell membrane come from. Back in the times of the simple light microscope, the cell membrane appeared as a simple line, for the resolution of the light microscope is quite limited. Yet, despite the limitations of the light microscope, Gorter and Grendel in 1925 performed a very interesting investigation using erythrocytes, red blood cells. They worked out the surface area of a red blood cell, and then they removed the membrane of those red blood cells. The data they obtained showed that the surface area of the phospholipids that came out of the cell membrane was equal to twice the surface area of the red blood cell. This led them to the conclusion that the cell membrane must be made up of a bilayer, a lipid bilayer. And so the idea of the phospholipid bilayer was born, with the membrane of a cell being made up of two sheets of phospholipids, each with their hydrophilic head and their hydrophobic tails. Soon after the work of Gorter and Grendel came the work of Davison and Danieli, and they examined the surface tension of the cell membrane, and they examined the permeability of the cell membrane, and they postulated the idea that based on the nature of the cell membrane surface, its surface tension, and the kinds of substances that were allowed to enter or not allowed to enter the cell, and they proposed that the cell membrane was not just a phospholipid bilayer, but a sandwich type model with the phospholipid bilayer sandwiched between two layers of protein. And at that time there was no evidence to refute such a model. In fact, in the 1950s, with advances in electron microscope technology, the evidence to support this sandwich model from Davison and Danieli became even stronger, with electron micrographs showing the classic railroad track with two dark lines on the outside to represent the proteins and two lighter lines on the inside. So the Davison-Danieli model of the cell membrane remained for some time, with its birth in 1935 way up into the 1960s. And then, with advances in microscopic techniques and the quality of electron microscope images, Singer and Nicholson were able to propose the fluid mosaic model. Using evidence from freeze etching and advanced techniques like fluorescent antibody tagging, today we realize that the cell membrane is not a sandwich model, but it is made up of transmembrane protein.